Yes, so what I want to do today is go through a bit more of chapter 11, where we talk about our membranes. And then at the end, I'll do a little bit of review of the um, study guide that we have there. So I guess I'll go ahead and start by sharing my screen. Okay, so we had left off discussing um, some lateral diffusion in the membrane. We know that for most organisms, except for a few very weird archaea, we have this membrane bilayer where you have phospholipids on one side and phospholipids on the other side, and their fatty acid tails interact with each other. We draw them nice and neat like this, but they're often kind of jumbled up. They're more in the liquid phase than the gel phase. We had discussed lateral diffusion. So if we look at any particular glycerophospholipid or sphingolipid or even cholesterol or anything that's in the surface in there, it can move sideways pretty fast. It'll zip along there, right? We had discussed FRAP as a mechanism for understanding how this movement for measuring this. Okay, so actually, I'm glad we're talking about this. I gotta make sure I have a good question there for you guys. I think, yeah, I might change one of my questions. Might be about FRAP. I know there's some FRAP questions in there, but I have a good question that really relies on you guys thinking. So, but anyway, FRAP is just about this fluorescence return after photobleaching. You basically coat the outer membrane with a fluorescent probe that will bind to the lipid surface there, and then you zap it with a laser to burn out some of that fluorescence, and then you just measure how fast the fluorescence returns to that area, and that tells you how fast we have our lateral diffusion through the membrane. Okay. All right. So we talked about that, and basically, we'll see that this moves pretty quickly, but there are membrane rafts. There are little sections in there. This picture of multicolored squiggles is basically showing one particular labeled photolip, one particular labeled lipid head group that kind of stays in one particular region until it jumps into another region. Okay, and then it kind of stays there and jumps into another one, so on and so forth. So there are little sections. Okay, it might be kind of like if you go to the park, maybe I'm going to go play soccer over here or have a picnic over here, but you're going to spend a lot of time in one particular little area. You could theoretically walk everywhere, but depending on what you're doing, or in this case depending on our particular protein. Oh, I hate that thing there. Sorry. um, so yeah, IT fixed my computer, but now every 5 minutes I get a McAfee alert. I swear I'm about ready to just ditch the whole WSSU IT stuff. Anyway, but we have these different regions in our membrane. Okay, so that's where we kind of left off last Thursday. So today we're going to talk about the converse of our lateral diffusion. We're going to look at transverse diffusion, and this is taking one phospholipid that's in here and flipping it over to the other side. So we either go from the outside to the inside or the inside to the outside. Okay, so we're going to talk about our trans bilayer. Basically flipping over here from, in this case, from the top to the bottom, as opposed to lateral diffusion, which occurs on a microsecond time scale. This takes days to occur. Uncatalyzed, this basically does not happen. It's incredibly rare. Um, and if you think about why that might be such a big difference in time scale, you can look at the fact that our phospholipid has this big honking charge on it. That phosphate group and then whatever's attached is polar. So trying to bring something polar through this hydrophobic membrane section here in the middle is not going to be very energetically favorable. So that's not going to happen very often. Okay. So if we want to actually flip something across that way, we have a series of enzyme, well, I won't say enzymes, proteins that catalyze that diffusion in there. Okay. So we often call them flipases. But we actually have flipase, floppase, and scramblase. Okay, so our flipase is where we're going to take one from the outside and move it to the inside. In flop A's, we're going to do the opposite. We're going to go from the inside and flip it to the outside. So if you look at the two on the left down here on the bottom, flip A's and flop A's, they both require ATP in order to have this happen. So we're going to couple the hydrolysis of ATP and our consequent release of a large amount of energy. We're going to use that energy to move this charged head group across the hydrophobic region and flip it to the other side. Or if we're going from the inside to the outside, we'll flop it over there. Okay, so flip A's. You can think of that as an I. We're going to the inside. Flip Ace is to the inside. Flop Ace with the O is to the outside. All right. We also have a third one called Scramblase. Okay. So Scramblase is going to actually result in two of them flipping simultaneously. All right. So we're essentially harnessing the work required to move one across there to move the other one across as well. All right. So Scramblase does not require ATP. It basically, counterbalances them so that we have one flipping and one flopping. Okay. All right. So that's what we end up with there. Okay. All right, so flip A's, flop A's, and Scramblase all relate to our transverse diffusion. Again, transverse diffusion essentially does not happen uncatalyzed. We need, one of these enzymes to actually move them. Okay, two of them, flip A's and flop A's, require ATP. Scramblase doesn't require ATP. It just moves two at the same time. Okay. This leads us into a discussion of membrane rafts. If we go back to that picture that, lateral diffusion where you have one that's kind of locked in one place and it kind of jumps the fence and sticks in their place in there. Our rafts are just the name we give to a cluster of particular lipids that have slightly longer than usual tails. All right, so usually we're going to have our sphingolipids in here, so a little bit longer, and we kind of fluff up the membrane space in there as well too. So we have this little area that structurally is a little bit different. If you think about maybe like a baseball field, the pitcher's mound is raised up, or I always think of when they would have NFL games at the same time that baseball season was still going on, and they had some stadiums that shared both, so you could see the dirt in there for the baseball and the sod for the football. So you'd have basically different little regions in there. And these different phospholipid components then make it easier to keep other things in there. Like maybe we want a particular series of proteins in there, whether they're GPI linked or whether we want a bunch of cholesterol to be in there, or various proteins like caveolin. We'll talk a little bit about that a little bit later on. But our rafts just kind of allow us to group things together. Okay. The other analogy that I like for this is like a community garden, where you have like this whole big plot that everybody can go there and plant stuff, but really you kind of section it off into like little areas. 12 by 12 area, you can plant whatever you want here, and then your neighbor can go over here and plant whatever he or she wants to, and they're a 12 by 12 area. So we have this kind of segregation of proteins in there based on these rafts, and they're going to allow us to collect certain things in there, and we'll see why that becomes important here momentarily. Okay, if we look at caveolin, okay, caveolin kind of looks like a little hair clip or pin, something like that. So you can see that it has some structural tension in here, and when caveolin proteins start to interact with each other, they enhance that interaction to increase tension. So they're essentially kind of pulling parts of the membrane down. So if we have a whole bunch of caveolins, we're going to make this cavity in here as we pull the membrane in. And so we essentially make this nice little bud. This can be useful for endocytosis or exocytosis as well. You can see here our electron micrograph showing us that we have these particular little caveola that are being pulled in here. And this is done through a physical tension of these proteins grouping in the surface that exert force to kind of pull it down in there to make that cavity. Okay. All right. If we didn't have those, if we took those proteins out, we'd essentially have a larger cell because, as opposed to making this little dip in there, we can put them all on the surface and it would just basically be a little bit bigger. But we do this for endocytosis. Okay. So we're inducing this curvature. Okay. So if we have this nice intrinsic curvature, very high density of positive charges on there, it can react with our phospholipid head groups. So we can see that we can actually put something on there and kind of pull this up as well too, so that we end up with our charge. We do something similar. In this previous picture, we have caveolin proteins that are embedding themselves in the membrane and kind of pulling down on it. In this case, for exocytosis, we have a protein that binds to the outside and kind of pulls up on it. Okay, and there's a particular bar domain in there. Yeah, all right. And these proteins play a huge role when we talk about fusion. Okay, so membranes can fuse with each other because again, they're just kind of. I always like to think of like soap bubbles. You blow some bubbles, you can take two bubbles and have them fuse together and make a bigger one. Or you can play like Fruit Ninja and chop up bubbles, and you get a bunch of smaller bubbles as well too. So you can have these membranes that can fuse and that can bud off from each other. We can see this with the Golgi complex. So we have vesicles that will leave the Golgi complex and travel to the membrane. And there they can fuse with the membrane and take whatever they have inside them and exocytose them. We can also bring stuff in, like with our caveolin, we have endocytosis. We can fuse our endosome with a lysosome. So we bring something in, fuse it with a lysosome, we can chew it up. If we think about viral infection, the way that works is, excuse me, our viral particle will bind and induce a kind of fusion in there, and it can release its viral DNA or RNA into the cell. We can look at the fusion of the sperm and the egg as well, gynegenesis. We can have the fusion of small vacuoles as well in plants mainly, and then we can even look at meiosis, separation of our plasma membranes in there. Okay. We're going to look at a particular example of one here. All right. So we're going to talk about some neurotransmitters. And in this case, we are going to show how these neurotransmitters can be released through the mediation of snare-type proteins. So this is one of these crazy things where we have, an abbreviation that includes an abbreviation that includes an abbreviation, so on and so forth in here. All right, so we've got a couple of snare proteins, and they call that because they're just going to basically kind of wrap around each other. We're going to see this in a little more detail coming up here. But our T snare is basically our target. Okay, so in this case, we have this vesicle that's released with neurotransmitters inside of it. It has vesicle snares on its surface. These are the ones in red. They're going to interact with target snares, the ones in blue. Those are going to interact, wind around each other, which will pull it closer. And then our Q snares are going to actually, be the ones kind of here in our snaps, I guess, snaps and snares, are the ones that are going to actually promote the fusion and the release of the calcium into the other cell. So let's look at this with bigger pictures, maybe. All right. All right. So here we have a neurotransmitter-filled vesicle that approaches a plasma membrane. Okay. The V snares and T snares are complementary, so they will bind to each other and they'll start wrapping around each other. Okay. The snaps are like once these V and T snares interact with each other, the snaps wind around them and pull them closer together. So this physically pulls the vesicle into contact with the cell membrane. Okay, so what we now have is hemifusion. Okay, we have this vesicle that is now attached to the membrane, but it still has a separate space from the cell itself. Okay, so as they make contact, just like our two soap bubbles, they make contact. We can make a bigger bubble. Our hemifusion, we're in contact. We can see that our outer membrane is the same, but we still have this separate point in here. If we continue to add stress through our snaps winding around our V and T snares, we'll pull these close enough together that we'll have complete fusion. We'll actually induce a pore, and this can widen up and release the neurotransmitter to the interior of the cell. Okay. right? And that's essentially what happens for, let's say, acetylcholine, I believe. Um, I think it's acetylcholine uses this mechanism in here. But key components, that initial T snare V snare interaction, snaps winding around to pull them closer, hemifusion where they're connected but still separate, and then finally complete fusion where our interior of our vesicle is now introduced to the cytosol and we can release whatever payload is in there into the cell. All right. Okie dokie. Okay. So that is an example of transporting something across the membrane, but we're going to talk about membrane transport in some more detail here. We're going to break it down into basically 6 types. All right. But we have a few definitions to go through. Passive diffusion, we just kind of pass across there. All right. So it's kind of like if you walk around one of the buildings on campus and test the doors, like, hey, this door's unlocked, I can get in. It's just passive diffusion in there. Okay. If say you go to the hospital or over at Bricks, I know some of you do research over at the PTC-RC, you have to have a little ID, and your ID card will allow you in. So that would be more an example of active diffusion. Or maybe you can have someone, if you forget your ID card, security can escort you in to whoever you're going to see or whatever lab you're going to. Okay, so passive diffusion occurs, but it's usually small non-polar molecules that will passively diffuse. One example would be a nitrous oxide that can pass through the cells. But passive diffusion of polar molecules is a lot more difficult because we have to have desolvation. You got your polar molecules that are surrounded by water in the aqueous medium. They have to get rid of that water and get into membrane. So there's this high activation barrier. That's not very likely to occur, kind of like our flip-A's and flop-A's. It's typically going to need to be catalyzed. So we can have facilitated transport that will allow an alternate diffusion. So we can have active transport and passive transport. And these involve the use of various proteins that are called transporters, or sometimes permeases, or sometimes porins. Okay. All right. So how do we get something across the membrane? The whole thing is it has to be energetically favorable. Okay. So if we're talking about passive diffusion, we're not spending ATP or not using some form of energy to get it across there. We can have 2 types of diffusion. We can have a concentration dependent diffusion. So if I have a high concentration out here in area C1 and a low concentration in C2, I'm going to have a gradient where things are going to kind of flow downhill. Okay, so I'm going to have a chemical gradient. Okay, and we'll flow that way. All right. We might also have a charge gradient. Okay, so here I might have a much greater positive charge on the right-hand side and more negative charge on the left-hand side. So I can see that my positive charges are going to flow to the left, my negative charges are going to flow to the right, and eventually we'll hit equilibrium. Actually, we won't. We're going to stay as far away from equilibrium as we can because of the dynamic steady state, but we do have this gradient here where charges can flow. So if you think of ions like chlorine or potassium or sodium, they can flow against these gradients, whether these are charge gradients or concentration gradients. When we talk about the generation of ATP and we can actually have an electrochemical gradient. If we're pumping protons, we're pumping charges across, and we're pumping it into a concentration, so that our protons will want to flow back into the mitochondrial matrix because it's both a lower concentration of proton in the matrix and it's negatively charged on the inside of the matrix. All right. Let's look at some of these terms visually here. If I have a polar molecule that is solvated, it's in solution in here, and it needs to diffuse across, we have to desolvate it, get it through the membrane, and then resolvate it. Okay. So without a transporter, this is going to have a very high energy barrier. Okay, because that desolvation effect takes a lot of energy, and getting a charged particle through this hydrophobic region takes a lot of energy. So what we can do is utilize a transporter, which may have some key charges in it. We'll see some pictures of this coming up here in a moment, that will allow, not so much desolvation, but an interaction with other charged particles that will allow it to flow through. So our transport mechanism is lower energy than just going directly across the membrane. Okay. So to summarize, we have 6 types of transport. Okay, the simplest one, the ones that everybody will be able to memorize easily, is simple diffusion. It just goes across the membrane, just forces its way through there. And usually, this is going to be very small compounds, non-polar compounds, and they're only going to go down diffusion gradient.
With the bags.
We can also have facilitated diffusion, where maybe these are polar, and so they can't get across on their own, but they're going to flow down an electrochemical gradient to get inside. Okay, an example of this would be our ATPase that we'll see in a minute. We can have primary active transport, where we're going against a gradient, but we're harnessing something like ATP to drive it against that gradient. So, we're turning on a little motor to kind of pump out the water, pumping water uphill in essence. So we're using a primary active transport. We're using ATP to do that. Secondary active transport, okay, is kind of, if we add another step to this, we use our primary active transport to pump something, against a gradient. So now we have, a higher concentration in one place, say our higher concentration out here. As that flows back in, so we pump this out, we let it flow back in, and it brings another ion with it. Okay, so our secondary active transport is driven by an ion moving down its gradient. Okay, so I might use primary to pump ions against the gradient. Secondary, I let them flow back in, and they bring something else in there. Okay. We can have ion channels. I know if any of you have worked with maybe Dr. Overholt, I think he looks at ion-gated channels, but there are ion-gated channels and ligand-gated channels. So you can either have a ligand that will bind and open it up, or close it and allow the ions to flow in. Sometimes you can have, just the charge. The charge builds up as you get a high potential across the membrane. It causes that to open up. Ions flow in, reduce the electric potential, and it closes. So I think of my gated channels as being like those trash cans you step on and the lid comes up, and you take your foot off and it closes down. And the last type is a little strange. These are ionophores. Okay, we have ions that have these big charges. Sometimes these are bigger than just ions, and we can't have them diffuse directly across there, but we can put them on the inside of something we call an ionophore, which has a hydrophobic exterior that can freely diffuse across the membrane. When it gets inside, it can release the ion in there and go back and pick it up. So this is going to be like our shuttle bus. I guess if you park at the stadium, you can take the shuttle bus into class, and the shuttle bus releases a student there, and then you can go back and pick up more and do the reverse when school's over. Although I guess nobody's parking there now, right? Okay, so those are our 6 types. Awesome questions coming up here, right? You can just see what the questions are going to be for exam 5. Why I like these last 2 chapters too. It's a little easier to study for, I think. Okay. So when we look at transport systems, besides our 6 types of transports, when we have porins or transporters in here, we can further break these down into what they actually do. So a uniporter brings one substrate straight across the membrane. So it's just one thing, brings it across there. That's all it does. It's a uniport. We can also have co-transport. Okay, we can break that down into symport. A substrate 1 comes across, and it brings substrate 2 with it. It's like, you're letting me in the club, you got to bring my friend in the club too. Okay, right. Antiport is, as one goes in, the other one comes out. So again, the club's too crowded, somebody leaves, somebody else can come in. Okay, there you go. Dr. Ebert and his club analogies. Woo! I'm so in touch with the kids today, right? I was like, what are clubs? We haven't seen anything like that since, March. Okay. But uniport, symport, and antiport. Also, nice, good multiple choice questions here. And our co-transporter, symport, and antiport, we got 2 things. The names are fairly reasonable there. Okay. So let's look at a specific example. Let's talk about the glucose transporter. I have props today too. This is going to be my glucose molecule. If we take a look at the sequence of our glucose transporter, we can see that it's going to have about a dozen transmembrane regions. These transmembrane regions are going to be primarily hydrophobic residues. You might have a few polar residues in here, and you'll see that the red ones, which are charged, are almost all in the aqueous media, either inside or outside the cell, except, bam, there is a charged residue right smack in the middle of the membrane. So that is not energetically. I mean, these two are kind of in there, but they're near the surface. That's not bad. If you've got one right in the middle, that residue is doing something. Okay, so this is going to help shepherd our glucose molecule across the membrane. And typically, you'll see a charged residue like that will be highly conserved. If you mutate that, you're going to change the function of your glucose transporter. So let's see how this works. Essentially, our glucose transporter kind of looks like this. Now we're drawing this, it's really 12 regions in here.
These hammers. Hold on. These hammers.
Hold on.
Hold on.
All right. I'll get right back to this. All right. So back to our glucose transporter. Okay. So it looks something like this. It's got a spot where glucose can sit in there. Okay. So our transporter is here. All right. It opens up like this, and it can basically just change conformations so that you have kind of like, I'd say maybe it's a little bit like the rotating doors in hotels. So it keeps the two sides separate, but it can open and transport things in there. So basically, our glucose can bind to the outside. We change conformation. Our glucose gets to the inside and gets released into the cell. Okay, and then when our transporter is empty, changes back to the other conformation, picks up another glucose molecule, and does the same thing. So it just kind of repeats that back and forth. Okay, so it's essentially allowing glucose to bind, probably interacting with that charged residue in the center there. And then once glucose binds, we see this conformational change to open up the inside of the cell, and glucose can fall off. And then when glucose falls off, we conformationally change back to the other conformation so that we can repeat this over and over again. Okay, all right. So you'll almost certainly see a question about glucose transporters. In this, I think we're primarily talking about the GLUT1 transporter. We can do something with the bicarbonate transporter as well too. This is an example of an antiport. So in essence, what we have here is carbon dioxide that we produce. I go work out, whatever, and I make a lot of carbon dioxide. I'm going to combine that with water to make carbonic acid. All right. So I'm essentially going to have this H2CO3. So what I'm going to do is break this down into bicarbonate, and I'm going to export the bicarbonate out as I bring chlorine in. Okay, so you can see I'm pushing a negative charge out this way, but I'm maintaining my charge balance because I'm bringing in a negative charge over here. Okay, so in my muscles, this is what's happening, pushing this out, bringing in our chlorine. I go to the lungs and have the opposite effect. Okay, so I can make I can bring in the bicarbonate and release the chlorine anions, so I can regenerate my CO2 and exhale that out the lungs. Okay, alrighty. So again, we have tissue or organ differentiation, but we have the same thing going on here. We have an antiporter that allows one negative charge to go out as another negative charge comes in. And then we just reverse that in the lungs so that we can exhale our carbon dioxide. And the neat thing is carbon dioxide, is a nonpolar compound, so it can freely diffuse across. Bicarbonate, because it is an ion, can't diffuse across and needs a transporter. Okay. All right. If we go back to our glucose example, we actually have, 4, 5, 6, several different types of glucose transporters. So, in order to efficiently get glucose where it needs to go, we use a variety of these glucose transporters. So maybe here we have, a GLUT2, which is just going to flow across a downhill. We're basically going to get glucose into the blood. That's going to get carried away to the muscles and go and do something like that. So we have this low concentration in our blood cells. So we're going to flow downstream here. On the other hand, when I consume my glucose, yay, Mountain Dew, lots of sugar in there. Um, in order to get that into the epithelial cells in my gut, it's going to flow through this glucose transporter. It's going to bring some sodium along with it as well too. Okay, so that's going to allow it to flow in here, basically flowing uphill against the glucose concentration, because my concentration of glucose in the epithelial cells is typically going to be higher than in the gut. It's also going to be higher than in the blood. So here I am using a symporter to get both of these in, and then I use a uniporter to get the glucose out of the epithelial cell into the blood cells. Okay? And you can see that I also have another an antiporter in here that will pump out sodium and bring in potassium. Again, so I can keep my gradient concentration of sodium in here low, so I can have more sodium that wants to flow in and bring more glucose into the epithelial cell. Okay, so here I have basically all three types of transporters: a uniporter, a symporter, and an antiporter that are all playing a role to help get glucose from the gut into the epithelial cells, into the bloodstream, so they can go wherever they need to go. Okay. All right, and two different types of glucose transporters, one that's a symporter and one that's a uniporter. All righty. All right. So active transport, we've got primary active transport and we've got secondary active transport. Okay. So just to, we kind of mentioned those in that one slide that had the 6 different types in there. So this visually is just going to help us show what those two are. So primary active transport, I'm going to essentially push my substrate against a concentration gradient by burning ATP. I couple the hydrolysis of ATP, gives me a lot of energy. I can push that solute uphill into a higher concentration. Okay. Secondary active transport is a little bit more complicated. Okay, I do the same thing as in primary active transport, but here I'm not worried about solute number 1. What I want to do is I want to get solute number 2, the green one, into the cell. So what I do is I push solute 1 out of the cell, and then solute 1 will flow back in and it'll bring S2 with it. Okay, so I use the energetics of S1 flowing downhill to I couple that with bringing 2, which is flowing uphill. Okay, so I can pull S2 in against the concentration gradient because I have enough energy from S1 flowing down a concentration gradient. But in order to establish that concentration gradient, I need primary active transport to pump out S1. S2 flowing in is a secondary active transport because it still requires spending ATP, but I'm not spending my ATP to bring in S2. I'm spending my ATP to set up my gradient that allows S2 to flow against the concentration in there. All right, damn it, damn McAfee. All right. So ATP hydrolysis will often drive the transport of substrates. We're going to see that in here. We can have these, if we want to transport nucleotides across the membrane, we have our nucleotide binding, sorry, if we want to transport anything across there, we have our nucleotide binding. We often talk about ATP hydrolysis, but it could be GTP or CTP or pretty much any nucleotide triphosphate. So we have those binding domains in there. If we look at something like this, we have our primary active transport. Here's our substrate. It binds. Our ATPs are bound to our transporter, and then our ATPs get hydrolyzed to induce that conformational change and bring in this substrate. Once the substrate passes through, our ADPs fall off. New ATP can bind. When new ATP binds, we get that change in conformation. So it's very similar to my glucose transporter, except now we're using a nucleotide triphosphate and the hydrolysis of that triphosphate to drive the conformational change. Okay. We can also talk about this in terms of diseases. If you think of cystic fibrosis, I don't know, I actually knew someone when I was in college who had cystic fibrosis, and ultimately she died of it because there's no cure or treatment. There are improving treatments, but it's essentially you're going to die. What happens is there's a particular a transporter that is regulated by this CRTF transporter protein. I think it's like a something, I forget what CR, regulatory transport something, in there. Um, but it's important for pumping chloride across the membrane. If you don't pump the chlorine across, then you don't properly hydrate the mucus that's in there, so you get this really kind of thick, sticky mucus that clogs up the cilia in the lungs, and so eventually the lungs end up filling with fluid. Um, and it's just an error in this particular gene that does not allow the transport of chlorine out there. Okay. So figuring out, understanding these various transporters can play roles in helping us find treatments or cures for various diseases. Of course, my favorite is always gene therapy. Let's just replace the bad gene with a proper gene. But gene therapy is not exactly easy to do. Okay. I always like to take a break and tell the story of when I first started grad school. I remember reading about, I was excited, there was a gene therapy clinical trial that was taking place. There were like 5 or 6 kids who had some terminal disease, and none of them had a prognosis of living more than like 4 months. They infected them with some particular virus that was engineered to carry in the proper gene, use reverse transcriptase to get it in there and treat it. Unfortunately, there was an unforeseen side effect, and they died. So whenever you kill kids, even if they're kids who are going to die in a few months anyway, if you kill kids, your clinical trial and your research area is going to get set back for a decade. There were no other gene therapy, like pretty much every gene therapy clinical trial, and there weren't many at that time, were shut down because, like, oh no, what happened? So again, the human body is immensely complex, and there's just something there that didn't quite work the way it was thought that it should. Anyway, um, back to our transport. We can also look at proton transport. This is a particular area that I really like to study because this takes place in the iron electron transport chain. Basically, you have a whole series of proteins that are geared towards pumping protons out of the mitochondrial matrix into the intermembrane space. Our mitochondria actually has 2 lipid bilayers. So this intermembrane space pump protons into there so that you have this huge gradient, very high concentration of proteins in the intermembrane space. And it also makes the intermembrane space much more positively charged. So you have this electrochemical gradient, and then you can allow the protons to flow down through complex 5 or the ATP synthase to drive the production of ATP. Okay. So essentially we have this ATPase that kind of looks like this, right? And it's actually a little motor. They're actually electron micrographs. You can actually kind of see this thing spinning through here. And there's a channel in here. I always think of like the gumball machines, right? You put your quarter in there and you crank turn the little thing there, and the gumball falls down and eventually kind of rolls out the other side. They do it that way so you can't like reach your finger up there and grab the gumballs and get a whole bunch out at one time. Um, but that's essentially what's happening here. We have these 6 subunits. It's this heterotrimer, basically, these 6 subunits that have a channel that will allow that will kind of rotate around. And as our protons move through here, this turns and drives the phosphorylation of ADP to ATP. Okay. Um, we also have something called aquaporins. They'll let water through there. So, if your cells need to hydrate or dehydrate quickly, look at aquaporins. A lot of ion channels will maintain gradients for active transport. So we want to be, either bringing in potassium or kicking it out. So we'll have certain transporters that will allow maybe like sodium to come in. We kind of saw this with our endothelial cells and our glucose transporter. We have another transporter that pumps that sodium out. So our ion channels are maintaining these gradients. Okay, and they do this by basically essentially surrounding our various ions, in this case potassium, to interact with that charge there too. Okay, so ion channels can respond to changes in voltage. Again, voltage gated ion channels. We essentially make a nice little lid there. We build up this electric potential as we pump charges across the membrane. Eventually, we get to a certain point where the potential is so high, our lid opens and we let ions flow in, and that reduces our electric potential. So the lid closes, we start pumping the ions back out, we build up that potential, and we open up the lid again. So we have this constant opening and closing to allow ions to flow in and out, in was it influx and efflux of ions. So this slide, way more than you need to know, but this just kind of gives some examples of various types of those 6 types of transporters in there. I don't think we really talked about our ionophores too much, and we won't talk about those too much, but that's basically just surround an ion, carry it across, and then release it. Okay. All right. I want to make jokes about that, but they would be improper jokes. Okay. So this chapter, like I say, is very nicely straightforward. We're just talking about our lipid bilayers. How do they form? What's their role? What are they composed of? And then, basically our transverse and our lateral diffusion of components through there, organization of it a little bit as well too. And then we can talk about membrane fusion and transport of solutes in there. So I think it's nice and fairly straightforward. And I'll pause for a second and let you guys ask any questions you have about this chapter. All right, if any. Okay. All righty. So then what I'll do is I'll pull up the study guide for the exam that is on Thursday, 10th, 12th. It's all running together at the end of the semester, right? Sorry to panic several people. I got like a bunch of emails all at once, like what exam? So sorry about that. It's my bad. My bad. All right. Let me share the screen here. Study guide. All right. Let's blow this up a little bit here. Okay. Is this the right one? Yes. Had to think of what chapters we're on. Man, it's been a long semester. Okay. All right. So this looks like a fairly long study guide, and I probably could have like gone back and edited out some stuff because I'm not going to ask you to draw structures, but I may ask you to know structures, particularly adenine, guanine, cytidine, thymine, and uracil. So know what the
Dr. Ebert,
yes.
the screen's blank.
Uh-oh. All right, let me try. Okay. Can you see it now?
Yeah.
Okay. That's an interesting thing to do. That always learning little things about here. I can't expand it up to blow up the screen. I was debating about trying. So I've got I'm at home today, so I've got multiple screens here. It's like, what happens if I throw this on another screen? It's like, what screen is going to get shared in Zoom? But I'll play around with that for next semester or something. Okay. All right. But yeah, so I'll just do it this way, and hopefully you guys can see all this. Right. Okay. There we go. All right. So the exam on Thursday is basically, there's a fair amount of structures in there. Only really 5. The rest of it's just kind of like mixing and matching. But we're really kind of talking about nucleotides. We've got our nucleobase in there. That's our adenine or guanine or cytosine or thymine, sometimes uracil. But just knowing the difference between the pyrimidines and the purines can kind of help you out there as well too. I think about this, I like the open-ended exam. I pretty much always ask people to draw adenine or adenosine triphosphate, draw an ATP molecule. But now with multiple choice, I'm like, okay, here's a structure. What is this structure? And so maybe I'll just have my purines there. Is it, adenine, deoxyadenine, guanine, deoxyguanine, same way I might partner up with C and T. So being able to distinguish between those different nucleobases will be probably important. The riboses are pretty much the same structurally. You've got that furan ring, that 5-membered ring with an oxygen at the top. The only difference is the 2' carbon is either has a hydroxyl group for for RNA, or it lacks that for DNA. And again, understanding that lack of a hydroxyl group contributes to the stability of DNA is a good thing to know. I don't do as much with the glycosidic bond in here. Normally I'd say draw adenine, draw another nucleic acid, now draw a connection between them. But maybe I'll ask you a question about this glycosidic bond. Again, we have this beta N glycosidic bond where we're going to have our nice little nitrogens that are going to interact with each other here. So we can talk about our our 5' to 3' connections between our polynucleotides. We'll also talk about this the syn versus anti as well too. Sorry, so the glycosidic bond is actually the nucleobase binding to the furin. Our polynucleotides, we can see that 5' to 3'. It's basically connecting our ribose rings to each other, and the nucleobases are all kind of off to one side. So if you like analogies, you can think of the nucleobases as like the side chains of proteins in a way. They're analogous that way, that they can go out and interact. Although that might be worse. That just depends on how your brain works there too. Um, I won't have you guys draw a phosphodiester linkage, but I might ask you, like, what does 5' to 3' mean, or what's going on here? We're going to hydrolyze an inorganic pyrophosphate, get rid of 2 of those phosphates, and leave behind 1 phosphate that is going to link the 5' carbon of one nucleotide to the 3' carbon of another nucleotide. So 5' to 3' stuff. Yeah, complementarity and understanding the sequences. If you have one DNA strand, you're going to have a complementary strand that runs in the opposite direction. So you're going to have 5' to 3' this way, and 5' to 3' this way. And in fact, I have a question that I love. I think I've asked it on every exam or some variation of it that deals with that complementary, drawing a complementary strand. And then understanding too how you get from mRNA to DNA or vice versa, I guess, if you're doing like a constructed DNA. I don't think I really ask about tRNA in this exam. And if I do, there's only like one little multiple choice question there. But mRNA certainly plays a big role. What happens during denaturization? We take our DNA, we melt it, we separate it out there, and it'll spontaneously reanneal. So complementary strands will bind to each other. That becomes important when you get into chapter 8 and say talk about microarrays or sequencing or something like that. You can have probes, or when we talk about cloning, you have your primers, they're going to bind to a complementary sequence. So if we take our DNA and melt it, there's all of a sudden a space for one of our probes to bind there too. So there's a lot of good stuff about definitions here, like what exactly is a plasmid or a transient transvection versus stable transvection. I think I do ask about that. Yeah. Talk about cloning. I think for the most part, there's nothing too crazy going on in here. What I've done with the exam is I think there's somewhere around 12 to 15 multiple choice questions. So not a terrible whole big number. But then I have I think like 4 or 5 or 6 short essay questions, and I have you guys choose like 3 or 4 of them. I think I've got like 4 or 5, and you choose 3, and then I have 4 longer essay questions, and you choose 3 of those, I believe. Yeah, I think that sounds about right. Okay, and like I say, the exam ends up being out of something like 104 to 106 points. I forget off the top of my head in there. So there's some built in, like there's one essay question that's only worth like 2 points. I'm like, okay, it's really worth like 6 or 8 or something. I can't remember how I did that, but I've got a little parenthetical note in there that tells you that this is really worth more. It's just that's the bonus points that are going around through there. Okay. All right. And I realized I probably started a little early today, didn't I? I was thinking we're starting a little. I wonder. That's a good thing I recorded this, isn't it? Yeah. All right. Questions about anything?
I just had a question. Did you figure out about the biochem too?
So I think by the end of this week, we should probably know. I know that there are at least 4 or 5 people who have said, yeah, we would take biochem too if it were offered. So now it just kind of goes to the chair and the dean. There is a chance because we found some alternative. I learned something new this month basically about the way like we have a bunch of professors who have release time for grants. So that release time includes money that should be accessible to hire people to teach classes. But the administration won't release that to us because we don't have enough chemistry majors. I'm like, that I don't know, that doesn't sound right. But I learned. It's kind of like when talk about politics and sausage making. You don't want to see how it's done. I kind of learned how this is done, so we're shaking our fist or something. But we found an alternative way to budget for this class. Um, like I say, it'll come down to the dean's decision. But if you haven't done that little survey, well, it's going to be in the exam as well too. But the more people who are interested in the class, the more likely that they'll lean towards it. But I know we've got probably like 5 who would take it at least. Um, so I'll keep you guys posted. Um, the other thing I'll say is, I know some students have had issues with conflicts in time. Normally, there's not a conflict, with biochem being offered the other classes that are in there. So, it's yeah. Well, we're trying to work through that, but it's not something I can do if I were in charge. Yeah, maybe in a couple years down the road. But yeah, we're working to make sure that we can offer what we can there too. And I know it was just kind of a surprise, but it was a surprise to me too when I looked at the course assignments for the spring, and I'm like, I'm not teaching biochem too. I thought Dr. Wu was teaching it for the first time in several years. I'm like, nope. It's like we're not offering it. Yeah. Meanwhile, students are like, hey, you probably not offer PChem, right? Yeah, ha ha ha. All right, but I don't know. Biochem two's it's a good class. Like I say, I completely changed my grading schema for biochem too. But anyway, I don't want to get trapped down that bunny trail there too. But like I say, I think we'll know something definite by the end of the week. It'll be something you can always add. I mean, it's never going to be full. I think the biggest I've ever had in biochem 2 were 12 students. So, and it's usually more like 6 to 8. So, but it is a very useful class. Yeah, we'll say that. Okay. Any other questions or? I know I've had some students ask about grades as well too. The biggest thing is I think I had to go back and check the first 2 exams to make sure that my alterations of the points applied the way I wanted them to. But as long as you're doing the homework, there will also be. Is it this chapter 9? It won't be till after the exam. So sometime next week, there'll be another little assignment that will show up. I believe it focuses on chapter 12 primarily, but it talks about cell transport and signal transduction, which is what chapter 12 is all about. So that will be a way to add some points in there as well too. For the most part, I'm not super concerned about anybody. Everybody is, participating and doing work. The big thing is making sure you take advantage of being able to do homework problems twice. Usually the homework average for the class is in the 90s. So that's what it should be. So make sure you get your points there, and that will help offset any of the exams. Okay. All right. Other questions? Like I say, I think this exam, this exam on Thursday is maybe a little bit more challenging than exam number 3, but you guys have more options. So if you don't know this particular topic, it's like, I'm not going to answer that question. I'll just choose these other 2 or 3 to answer. So I think that helps everybody out a little bit as well too. Historically, the exam average for exam 4 is almost as high as exam 3, just a few points lower. Definitely exam 2 is the worst one, so we're through the worst part. Okay. All right. Other than that, I'll let you guys go. I'll post this lecture a little bit later on today. Now I got to go back and see if my son can get into his classes. Technology. We both have IT support tickets to different places for different reasons. Okay. Other than that, you guys have a good week. Oh, yes, question?
No, I was just gonna tell you to have a nice day.
Okay. Thank you. And hopefully I'll see you all in person sometime before the end of the semester. Take care.